This is the set of lecture slides for Unit 1, Chapter 4 on bacteria and archaea. So figure 4.1 in Chapter 4 in the textbook has an image of a bacteria cell, and it identifies major structures in the cell. I've highlighted the structures that we'll focus on in this class and that you'll be responsible for on your exam with the purple boxes. So when you're preparing for the exam, going back to figure 4.1 and reviewing all of these structures would be a good way to make sure you have a handle on the parts of the cell that you're responsible for. So let's start by looking at bacteria cells overall and discussing different ways that they can be shaped, different morphologies, and different ways they can be organized or arranged. So remember, bacteria are unicellular organisms. They are prokaryotic cells that do not have a nucleus. And although they are unicellular organisms, bacteria can associate together into groups with other single-celled bacteria. And bacteria that work into these groups or arrangements are of the same species. So all bacteria have a morphology or shape. Not all bacteria have an arrangement. Bacteria that don't have an arrangement would just exist as one cell without associating with any other cells. So for the morphologies or shape possibilities in bacteria, we have bacteria that are rounded, shown in Part A of Figure 4.3. These bacteria are described as coccus or cocci for plural. Bacteria can also be oblong or rod-shaped, and they are described as bacillus bacteria, or bacilli is plural. In category C, we have bacteria that are shaped vibrio, so their morphology is vibrio, where they are curved to shape like an L or a C. The spirillum and the spirochete bacteria are similarly shaped like corkscrews, with the spirochete having a tighter turn and a slightly longer cell size. Bacteria that have the morphology of a branching filament grow in a similar pattern to fungi in that they resemble branches on a tree. So shown in Part F of this figure, the drawing is two separate branching filament bacteria cells. So as I said, all bacteria have a morphology, be it coccus, bacillus, vibrio, spirillum, spirochete, or branching filament. Not all bacteria have an arrangement. So only bacteria that have an arrangement are the coccus bacteria. They can be associated into pairs, so two rounded coccus associate together into a group called diplococci. So their arrangement would be diplococci. Bacteria that have an arrangement of streptococci are rounded in shape, and the strepto tells us that they are arranged end to end in a long chain. So when they're cocci bacteria arranged in a strepto arrangement, they resemble pearls on a string. The staphylococci are bacteria that are rounded, and their arrangement of staphylo puts them in association with several other cocci bacteria into a cluster that resembles a cluster of grapes. The other bacterial shape that can have an arrangement are the bacilli. So the rod-shaped bacillus can be arranged as diplobacilli, so two bacilli cells arranged end to end, or streptobacilli, so this would be several bacilli bacteria cells arranged end to end. And shown in the electron microscope image in Part B are streptobacilli. So there are several discrete individual bacilli cells arranged end to end into a long chain of streptobacilli. There are several key differences between bacteria and human cells that we're going to discuss. The first being that bacteria are much smaller than human cells in size. Secondly, that human cells are eukaryotic and have a nucleus, where bacteria cells are prokaryotic and do not have a nucleus. In the eukaryotic cell, the DNA is organized in the nucleus, which is a membrane that surrounds the DNA and separates it from the rest of the cell. In the bacteria cell that is prokaryotic with no nucleus, the DNA is present in the cytoplasm and is in contact with the rest of the cell. Additionally, the DNA in bacteria is circular, and the DNA in the nucleus of a eukaryotic human cell is linear. Third, human cells have many membrane-bound organelles, and we'll discuss major functions of organelles on the next slide. The human cell has more organelles because the size of the cell is larger than the bacteria cell, and so volume has to be partitioned off to allow for reactions and functions to occur more quickly. The bacteria cell, on the other hand, is smaller in size, so does not require membrane-bound organelles. The major organelles in the cell that would be present in human cells are nucleus, ribosome, endoplasmic reticulum, and mitochondria. And the function of each is defined here. In the bacteria cell, which has fewer membrane-bound organelles because of its smaller size, there is only the ribosome, which is the site of protein synthesis, present. So a bacteria cell has a ribosome, and a human cell has a nucleus, ribosome, endoplasmic reticulum, and mitochondria. Key difference number 4 is the size of the ribosome. In the prokaryotic bacteria cell, the ribosome size is described as 70S, which is a unit called the Svedberg unit. The Svedberg unit is not additive, so 50 for the large subunit of the ribosome and 30 for the small subunit adds up to 70. In the eukaryotic cell, the ribosome size is 80S. So the eukaryotic human cell has a slightly bigger ribosome than the prokaryotic bacteria cell. The last key difference between bacteria and human cells is that bacteria have a cell wall, and this cell wall is made of a molecule called peptidoglycan. Human cells do not have a cell wall. Peptidoglycan is a molecule whose structure is shown in the bottom portion of figure 417 from your book. Generally, just know that it is a long chain of sugars that are cross-linked together to form a woven structure that gives the bacteria cell a lot of 3D shape and resistance to volume changes. When looking at the cell envelope of a bacteria cell, which would be the plasma membrane and the cell wall together, there are two categories of cell envelopes. Bacteria can have either a gram-negative cell envelope shown on the right in this figure, or a gram-positive cell envelope shown on the left in this figure. Major features of the gram-positive cell envelope are that the peptidoglycan cell wall is very thick, which is the purple layer in this image. There is a single plasma membrane, which is the yellow layer in this image. And there are lipoteichoic acid interwoven in the peptidoglycan cell wall. The gram-negative bacterial cell envelope, major features are a very thin layer of peptidoglycan cell wall, which is fuchsia on the right of figure 415. There are two plasma membranes, an inner plasma membrane and an outer plasma membrane. And extending from the exterior of the outer plasma membrane is a molecule called lipopolysaccharide, or LPS. Additionally, the gram-negative bacteria cell envelope has porin proteins, which are tunnels through the outer plasma membrane into the space between the outer plasma membrane and the cell wall. Because the gram-negative cell envelope has extra layers in comparison to the gram-positive cell envelope and porin proteins, gram-negative bacteria are difficult to treat with antimicrobial drugs because the antimicrobial drug has difficulty entering the interior space of the cell due to extra layers and the porin proteins. An easy laboratory test can be done to distinguish the gram-negative and gram-positive cell called the gram stain. This procedure is explained in this figure, and I will let you read this on your own and email me if you have any questions. As a result of the gram stain, we have gram-positive cells being purple when all of the steps in the gram stain protocol are done, and the gram-negative cells being pinkish red when all the steps in the protocol are completed. So visually, it's a very easy way to distinguish a gram-positive from a gram-negative bacteria cell. We'll now just talk about some extracellular structures in bacteria. So these are structures that are outside of the bacteria cell wall and cell membrane. So one of these is the glycocalyx, which is not present in all bacteria. So some bacteria have a glycocalyx, and the glycocalyx can be either a slime layer, shown on the left, which is a very watery, loose layer that is outside of the cell wall and cell membrane, or the glycocalyx can be capsule form, which is a more compacted, tighter layer. So in both of these glycocalyx drawings, the slime layer and the capsule are light brown or tan. Having a capsule for a bacteria is very beneficial because it can help evade immune defenses in a host. So if a bacteria with a capsule infects a human, the human's immune system will respond by sending phagocytes to kill the bacteria cell. But a phagocyte has difficulty attaching to a bacteria cell with a capsule. So in this image, the phagocyte attempts to capture the bacteria, but because the bacteria has a capsule, the bacteria escapes the phagocyte. Capsules are also beneficial in helping bacteria form biofilms because they help bacteria stick to surfaces. Once they stick, they can secrete capsule deposits, and they then form a layer of protection that is hard to penetrate with antibiotic drugs, sanitizing agents, and cleaners, and is very difficult to remove. These are a big problem in hospital environments in places like catheters and other surfaces where body fluids and bacteria are present. Some bacteria have extracellular structures that allow them to be motile, meaning they can move. A bacteria can either move at random, as shown in part A, or can move toward a chemical attractant or some other sort of attractant in a process called chemotaxis. Bacteria move in a tumble and run fashion where they separate their structures for motility in the tumble. They bring them together in a run to propel them in one direction, and then they separate these structures again in the tumble. Fimbriae and pili are two extracellular structures that also help bacteria attach and form biofilms. Additionally, pili allow bacteria to connect, as the bacteria are in this image, and they can perform conjugation, which is sharing DNA between unrelated bacteria. And we will discuss this further in later lectures. Lastly, some bacteria have the ability to form endospores, which can help them to survive adverse conditions like depletion of nutrients. Inside of an endospore is DNA sometimes RNA, ribosomes, and enzymes that are all important for the bacteria. The spore, however, is not metabolically active. When the adverse condition has passed, the bacteria can then become a normal vegetative cell and exit the spore state, and then can start doing metabolism, can divide, have daughter cells, and will then be alive. It has been shown that bacteria endospores can survive thousands of years, extreme heat, dehydration, toxic chemicals, and radiation. So that concludes Chapter 4. Now that you've watched Chapter 1 and Chapter 4 lectures, you can complete the handouts, and then you'll be ready to do your first homework.